My name is Andrea Ullius. I work as a scientist at the R&D at QIAGEN for the pre-analytics. It's a joint venture between QIAGEN and Becton Dickinson. I develop products for sample collection, stabilization and extraction of nucleic acids for standardizing pre-analytical workflows to enable reliable testing results. At AACR, I am presenting a poster containing data of a collaboration partner from the University Clinic of Halle and from the DRK clinics in Berlin. Professor Schmidt and Dr. Fleischacker provided data of an ongoing research study which tests the application of the PAXgene blood ccfDNA tube for monitoring therapy outcome of late-stage lung cancer patients. We were included in the pre-analytical part of the project, so it was very interesting for us to work together with a collaboration partner that has access to patient samples, and it's a clinical study. So normally we develop products based on healthy blood and healthy donors, and in this case it was now in a clinical setting. It's a research study, but still it's a clinical setting, and it was very interesting for us to see how our product can have an impact on monitoring cancer in a very early phase of therapy. The study was divided into two parts. In the first part, they investigated whether the PAXgene blood ccfDNA is applicable for their workflow, in which they formerly used the EDTA tubes, and whether you can store the samples for a longer time period compared to the EDTA tubes. And that was very successful. Both biomarkers could be analyzed as good as with EDTA. In the second part of the study, they used the PAXgene blood ccfDNA tubes only for monitoring therapy outcome of lung cancer patients, and they compared the results of the monitoring with the conventional restaging of the patients, and it could be shown that with blood sampling or with using of liquid biopsy, an earlier assessing of therapy failure or success could be possible for these patients. I had a lot of traffic. There was a lot of interesting scientific discussions at the poster. There were many interested clinicians as well as basic researchers, and there was a lot of interest in our tube especially.
QIAGEN.
Sample to insight.